Good morning, everyone. I'm really excited to be here. I'm a third-year PhD candidate at the Stowers Institute for Medical Research, and I'm really excited to tell you about this part of my PhD thesis focusing on epigenetics in zebrafish ribosomal DNA. And I'll start with a brief introduction. And we study zebrafish, this fish that I have here, which is a teleost, and I'll be talking more about that in a few slides. And this particular fish displays a remarkable developmental switch in the composition of the ribosome, the machinery responsible for protein synthesis. And as we can see here in this simple schematic, the ribosome will start in early development, having this pink identity, and then throughout development it will change to this blue ribosome. And specifically, the component that is changing over time are the ribosomal RNAs, which represent the catalytic component of the ribosomes, meaning that they will form the peptide bonds during translation. And then these pink ribosomes that I was showing you before are the maternal ribosomes that are made up of these maternal rRNAs. And then this blue ribosome are the somatic ribosomes made up of somatic rRNA. And a very important feature about these rRNAs is that they share a 90% sequence identity, which seems quite high, but for ribosomal RNA, this is very different. And to put into context, 90% of sequence identity at the ribosomal RNA level is usually seen in species that span hundreds of millions of years of evolution. However, we have them in the same genome in zebrafish. And the maternal ribosomes are called maternal because these rRNAs are primarily expressed during oogenesis, and then they will be maternally deposited in the eggs. And then, as I was saying, throughout development, these ribosomes will be replaced by the somatic ribosomes made of these somatic rRNAs that will start expressing after zygotic genome activation, and it will be present in virtually all adult tissues. So I wanted to show this expression map. So I color-coded a male and a female with the expression of these two subtypes of rRNAs. So we see that in the male, it's all blue because all tissues express the somatic rRNAs, and then in the female, we only see the maternal in the ovary that will later be deposited in the eggs, and those ribosomes will carry the first few stages of development. And even though there's 4 rRNAs that make the ribosomes, for today's talk, I will be only focusing on these 3 that are encoded in the same transcriptional unit, the TRM45S rRNA gene. And these genes are special because they are organized in highly identical repetitive arrays. Each of these transcriptional units encoding these 3 mature RNAs are usually 10 kb, and there's hundreds of copies of these representing hundreds of kilobases of DNA to even megabases of DNA. So these arrays are really massive. And this repetitive nature and the high identity across the repeats makes them really difficult to assemble with short-read sequencing data, meaning that those genomes that were assembled with short-read completely lack ribosomal RNA references. And in zebrafish, these 2 subtypes of 45S genes exist in separate arrays, and that's what I'm trying to show you here with this figure. So the maternal array is in 1 chromosome, and then there's 3 arrays of somatic in 3 other chromosomes. So these are not alleles. These represent fully different arrays that are encoded in this spatiotemporal manner. So with all of this introduction, there's 3 questions that we think are important with this particular system. And one is whether there's a functional consequence in these ribosomes. Because the composition is different, then this opens the possibility for different translation capacities. And in fact, this is a question that a lot of translation groups are working on. But for today's talk, I will be focusing on these other 2 questions. What's the evolutionary history? How do you end up with these separate arrays of these genes that are so divergent within a single genome? And the second part, what's the regulatory mechanism behind this switch across development? And I'll start first with the evolution history, and this will be a little bit of a snippet of what we've been doing here. And as I was saying earlier, zebrafish is a teleost, which is a group of fish that originated with a whole genome duplication event approximately 350 million years ago. And this represents the most diverse group of vertebrates, encompassing more than 26,000 known species. So we reasoned that because there's this whole genome duplication event in teleosts, and zebrafish is a teleost, maybe these 2 arrays originated with this specific event. And with that reasoning, then most teleosts should have these 2 arrays of 45S genes. So we started to address this by looking at publicly available short-read sequencing data of more than 1,500 species to see if we could find this evidence of having these two subtypes of 45S genes. And to make a really long story short, what we found essentially is that most teleosts show one single kind of 45S gene similar to all the models we know, and it's only Zebrafish and its close relatives that show this particular presence of two different arrays of 45S genes. And we were able to validate this with long-read sequencing because, as I was saying earlier, short-read sequencing gave us an answer of maybe which species could be good candidates, but we really needed a long-read to validate that these different sequences existed within a single genome. And we did this with PacBio long-read data that we generated in-house at the Stowers Institute, and we validated that Zebrafish, represented here in the Daño genus, has these maternal-somatic sequences. We also see it in the Vario, which is the sister genus to Zebrafish, and then in this close related fish, Dañonella, I'm showing a gray ribosome representing a single kind of 45S gene, and similar in this other outgroup, Astyanax. And this led us to believe that the origin of this particular system of maternal and somatic switch originated in the common ancestor to Daño and the Vario. So as I was saying, this part was a little short, and what I talked about today is that PacBio allowed us to confirm the sequence identity of 45S genes of a number of teleosts, and we're still sequencing more species. And our data seems to point that Zebrafish 45S subtypes likely originated in the Daño and the Vario common ancestor. And I wanted to start first with this evolution history to leverage comparative genomics to explore the mechanism behind the maternal and somatic switch in Zebrafish. Ribosomal RNAs are essential, so we cannot knock them out to see what functions they might have. So we needed an alternative approach to dissect the mechanism, and we think that comparative genomics, by having a number of species with the system and without it, is the answer to that particular question. So then, now for the main point of my talk, I will be talking about the epigenetic regulation and hoping to understand what are some of the features that are driving the spatio-temporal expression of these two kinds of ribosomal RNAs. And I wanted to start first with a little introduction of how 45S transcription is regulated. And one essential part to this process is binding of a transcription factor called UBTF that will bind to the promoter region of these genes, will recruit the transcriptional machinery to drive transcription. And then working done in mice revealed that DNA methylation in the context of a promoter can prevent UBTF binding and then will block transcription. And this led people to believe that UBTF is incapable of binding methylated DNA. And our lab has contributed to this story even further by looking at primate cells. And I'm going to show you a simple example of what our lab has found, specifically in human cells. And in this cell, there's arrays, as shown here with this picture, that will be either active or bound by UBTF, or inactive or not bound by UBTF. And with this particular system, our lab was able to define some of the epigenetic features behind these active and inactive arrays. And in fact, the active genes show really low CpG methylation or DNA methylation, and then the inactive genes show higher DNA methylation. And this also correlates very well with chromatin accessibility, where the active genes are accessible and the inactive genes are inaccessible. So with all of this information that was generated in our lab, we were really interested in exploring how this looked like in the Zebrafish system specifically, because this genome needs to accommodate these two kinds of arrays. So we wonder if CpG methylation could be a predictor for 45S transcription in Zebrafish. And to address this question, we started looking specifically at the early embryo with publicly available short-read sequencing data, and this was by sulfide sequencing. And we started plotting the average DNA methylation across these two genes, or these two kinds of genes in the early embryo. And as we can see here, the somatic is really high, maybe consistent with not being transcribed at these early stages. And then the maternal DNA methylation is very low, consistent with this array being active in these stages. So we reasoned at the time when we had this publicly available data that what we were going to see, if we kept measuring across development, that the maternal will keep going up to be fully silenced, matching what was published before, and then the somatic methylation will go down to be actively transcribed later in development. So at this point, we started generating our own data, and we added 3 more time points to this particular plot, which are the 24-hour, 72-hour, and 120-hour points in hours post-fertilization in the zebrafish development. And as expected, the maternal kept going up, consistent with not being actively transcribed, but then the somatic was not going down. It kept being highly methylated in all of our measurements. And we had also RNA-seq data matching these time points, and we saw that it was actively transcribed despite being highly methylated. So we reasoned that maybe this was an artifact or a phenomenon specific to early development, because early development regulation is slightly different than the homeostatic adult tissue. So we decided to look in the adult as well. And we look at 5 different tissues in males and females: muscle, heart, gut, fin, and gonads. And in all cases, we saw that the somatic was highly methylated, the maternal was highly methylated in all of these tissues where it was not expressed, and only was lowly methylated in the ovary. And as I was saying earlier, we matched all of these samples with ribosomal RNA sequencing data, and we see indeed that the somatic is expressed as early as 6 hours, and then throughout development, and fully expressed in all the adult tissues represented by the text highlighted and the check marks. So to further characterize what was going on with this system, with this really surprising result of a gene being actively transcribed despite being highly methylated, we decided to use the 24-hour time point in the early embryo as a model to further study what was going on in the system. So as I was saying earlier, we did ribosomal RNA sequencing with all of these time points, and I wanted to show you the data for the 24-hour embryo. And we measure rRNA in 2 different ways. One called nascent, where we're mapping reads to this spacer that will be quickly degraded after transcription. So if we see any reads mapping to the spacer, that means that that was recently transcribed. And then total rRNA matching to the mature rRNAs, representing the pool of nascent rRNA and those RNAs incorporated to the ribosome. So when we look at the total RNA, we see that at this stage, the majority is still maternal because it's really abundant in the early embryo. But when we look at nascent, what is actively being transcribed, almost 100% of it is of somatic origin. And there's a caveat of everything I've shown you so far, which is that we were using short-read method sequencing, which, as I said earlier, short-read is very difficult to map to these sequences, and it's an indirect measurement of DNA methylation. So we were wondering if what we were measuring was indeed an artifact and not really biology. So we needed to resort to a different technology to be able to fully address this question and understand what was going on at the epigenetic level. So we resorted to long-read sequencing on native DNA with PacBio to detect native DNA methylation with single molecule resolution. And what I'm showing you here is the average methylation across the repeat of the somatic 45S at the 24-hour embryo. In the first track, we see the methyl-seq data that is really high, as I showed you before. Then we use 2 long-read sequencing methods to validate that this high methylation was real. We use nanopore and we use PacBio as well. And all 3 orthogonal methods that measure DNA methylation in very different ways agreed that the methylation was quite high. And when we were doing these experiments last year, that's when PacBio announced a new chemistry that improved DNA methylation calling, and we were really interested in trying that. So we got the reagents, we got the flow cell, and we resequenced this PacBio library, the exact same library with a new chemistry, and we indeed see an improvement in the DNA methylation calling, because now with the new Spark chemistry, PacBio methylation calling closely resembles the field standard, which is methyl-seq. So we were really excited about this new improvement, and we moved forward with these experiments with this new chemistry. And as I was saying in a couple slides ago, long read gives us the ability to look at single repeat resolution. So I'm showing you here a bunch of reads. Each line here represents a read that is color-coded by the DNA methylation, being methylated in red and non-methylated in blue. And then we see that the majority of the reads are red, and maybe we see 1 or 2 reads that are blue, consistent with low DNA methylation. But this was with the old chemistry, and we resequenced the same library with a new chemistry, and we see that those reads that show low methylation are completely gone, and now the majority of the reads are highly, highly methylated, which is quite unusual because ribosomal RNA is the most abundantly transcribed locus in the genome, and then it's highly methylated but being actively transcribed. So another feature that we wanted to address with this, because we couldn't use DNA methylation as a proxy for transcription because there's something going on in this system, we resorted to the transcription factor I showed you before, UBTF, wondering if it is capable of binding methylated DNA in zebrafish. So we addressed this first with an imaging approach, and I'm showing you here 3 different images. So we combine fluorescence in situ hybridization with immunofluorescence. So we have this universal 45S probe that binds both the maternal and somatic arrays, one specific to the somatic 45S, and then UBTF, and we see a perfect overlap, which shows us that UBTF colocalizes with somatic 45S despite being highly methylated. But this particular approach is just measuring colocalization. It's not necessarily measuring direct binding, which is what we address next. So we perform Canon-1, which is a protocol similar to ChIP-seq, where you direct antibodies to your protein of interest, and then you cut the chromatin surrounding where the antibody is bound, and then you sequence those fragments. And because we were interested in addressing whether UBTF was bound indeed to methylated DNA, we sequenced this library with nanopore to be able to detect the native methylation from the fragments coming from these experiments. And I'm showing you here the data. So we have the reads mapping to the maternal and somatic 45S, and we indeed see with both replicates that UBTF is bound to the somatic 45S, and then it's indeed bound to highly methylated DNA. So at this point, we were wondering if these unusual methylation patterns that we were seeing in zebrafish were specific to zebrafish to accommodate for the maternal and somatic arrays, or maybe it was an artifact, not an artifact, or maybe it was something more broad that was occurring in other species. So this is the point where we came back to our comparative genomics approach, and we look at the DNA methylation of the other species that we had in our analysis. So I'm showing you here this tree showing the 4 genera that we're looking at. And first I'm showing you the data for the Vario, this closely related species to zebrafish that has the maternal and somatic switch. And then we also see these really high DNA methylation levels, which is very similar to what we see in zebrafish. But we also have these R groups, systems that don't have the maternal and somatic switch. And even though in Danionella we see a few more reads that are non-methylated, we still see that the majority are abundantly methylated. And we see something very similar in our other R group, Astyanax. And I wanted to show you here how a mammalian system looks like to make the point, the strong point that what we're seeing is really unusual high methylation levels. So when we look at the mouse and we look at the reads mapping to the 45S, we see that the majority of those reads are fully hypomethylated, and there's only a handful of reads that are highly methylated, which is the opposite of what we see in teleosts. So I wanted to summarize the second part by briefly mentioning that the maternal 45S does match what we see in mammalian systems, where the methylation inversely correlates with expression, high methylation not expressed, low methylation expressed. But then the somatic 45S is a completely different story because it's constitutively hypermethylated despite being actively transcribed and representing about 80% of the total rRNA, or the total RNA in the cell. Another interesting finding is that UBTF binds methylated 45S genes in zebrafish, which is opposite of what happens in mouse and in mammalian systems in general, which supports a new mode of 45S regulation. And we observe as well that high levels of 5-MC methylation of these genes might be a common feature of teleost. And I wanted to make a point here that everything that we know about chromatin regulation does not apply to the 45S genes of zebrafish. And these are findings that are being powered by long-read sequencing technology, specifically with PacBio. So we're wondering, what else are we missing just because we didn't have the technology to look? But now we have the technology, so we have a responsibility to start looking to see what kinds of biology we were missing. And in this case, I want to make the point again that this is the most abundantly transcribed loci of the genome, despite being highly methylated, which is completely opposite to everything we know about chromatin and gene expression regulation. And I wanted to wrap up with a few future directions, some of the things that we're working on. A nice outcome of using long-read sequencing data is that we can use it to assemble genomes. And in fact, we've worked on assembling a near T2T, a near telomere to telomere genome for this fish, the Vario, and we're almost done with this, so this should be coming out soon, and we're really excited about this. We're also working on implementing fiber-seq which is a proxy for chromatin accessibility with long read, also powered by PacBio in Zebrafish. And we're also working on further functionally exploring the role of DNA methylation in the rDNA regulation of teleosts, because as I said, all the rules that we know that apply to chromatin don't apply to Zebrafish, so we need to really start writing the new rules of this particular DNA. And with that, I would like to thank my lab, specifically my PI, Jennifer Gerton, for being extremely supportive with this project. All the Stowers core facilities. We have here with us Anosha, Kate, and Sean, who have been doing all the sequencing that I show you today, my committee members, my collaborators, the organizers for giving me the opportunity to share with you today some of my PhD project, and all of you for listening. And I'll be happy to take questions. Thank you.